Yep, and we're coming up to our 9 weeks test, and oh boy, it's in Test Media Center B, and you'll come to the door, and you're just supposed to meet me over there. It's going to be 30 some odd questions. You're going to be on a computer. Away you go, and it'll be your 9 weeks test worth 20%. So it is going to give you a big bump. Look, the next question on the test it deals with something we've talked about a bunch. It is talking about the selectively permeable membrane. And kind of what does it do? You know, it does control what molecules can get in and can get out. But I don't know if I ever mentioned that the selectively permeable, the membrane itself, it does another thing. It also helps to mark the cell. By that I mean the proteins that are sticking out of the cell that provide for the channels for these molecules to go in and out. The body of a multicellular organism, especially of humans, that kind of thing about humans, can recognize that that cell belongs to me, belongs to self. So let's not attack that cell, because the protein markers are so familiar. And it also, the membrane will also protect the cell, certainly not against radiation, but it would protect it against unwanted molecules, and it would protect it against a certain amount of heat, certain amount of cold. You know, I mean, now we're not talking about frostbite cold, we're not talking about, open flame heat. But it can adjust to external environments, can protect it against unwanted chemicals. The next question talks about this movement of molecules through the cell membrane. And, we had a couple of them. One was diffusion, and when you're talking about diffusion of water, we talk about osmosis. Another one talked about facilitated diffusion and active transport, and those were two others. And there was endocytosis and exocytosis, those as well. But as far as the facilitated diffusion, that's one where some structures are built in the membrane that act like special gateways for certain molecules. So the proteins, the structures, the proteins in the membrane fit and invite and help to facilitate, meaning to help happen the movement of certain molecules. So they're shaped in such a way as to attract them. Now, active transport, a little bit different. Active transport, typically, you're going to go against a concentration gradient.
Mr. Hernandez, please report to the IB office for your conference. Mr. Hernandez, thank you.
It's Mr. Hernandez. I'm glad I didn't say Mr. Stevens. I had a couple this week already, so, but anyway, active transport, like it says active, do you think it takes energy? Let's think. Active. Yes, it takes energy. So that's a big difference there. The facilitated diffusion, you could argue and say that it took energy to build the protein, but in the actual movement of the molecule, the structure of that built protein gives you the advantage. And in active transport, energy is actually expended to move that substance there. Now, let's talk a little bit about microscopes. I've got a couple different kinds. I mean, the first one is a light microscope. What can you see with a light microscope? Well, you can certainly see cells and the cell parts. You're pretty good at that. Tiny organisms, sure, you can see paramecium, amoeba, stuff like that. Can you get down to atoms? Put a big no on that. Not to atom size, okay? You can get to organelle size. You can get to cell. You can get to small organisms, but not to atom. Golly, not to molecules either. So keep that in mind. You want to go down to the atoms and to the molecules, you better go get yourself an electron microscope. There's a couple of different kinds, and they're starting to magnify things 400,000 times.
Ms. Rosenberger, please report to the IB office.
She needs to get to that IB office too. I don't. So, light microscopes, organelles, cells, tiny organisms. Now, when we're talking about the cell theory, we know that there are three parts to cell theory. Let's see. Number one, all living things are made of cells. Number two, cell is the basic building block of life. Part three, all cells come from pre-existing cells. Yeah, there's your cell theory there. And there you go. If you can remember that, you can get that question. Let's see. Now, let's talk about bacterial cells. You know, when you talk about bacterial cells, the big thing that'll jump out is like, wow, they are prokaryotes. Now, prokaryotes, what are those? Well, those are cells without a nucleus. So a bacterium, no nucleus. It's got some genetic material, but it's loosely arranged in its, and I want to say cytoplasm, I think that's what you would call it. But there's no nucleus, no mitochondria. You will find ribosomes in there, but ribosomes are considered microscopic bodies. They don't have a plasma membrane. So when you're talking about the bacterial cells, prokaryotes, there's not a nucleus. Okay, good. Oh my gosh, really? Yes. This next question has to deal with, you find yourself wondering what type of cell it is. Listen, if you find chloroplast in the cell, go ahead and say, plant cell. Go ahead and say plant cell. You could maybe argue some sort of protozoa, but don't worry about that. If you find yourself a cell with mitochondrion, you've at least moved into the eukaryotes. So if we're talking eukaryotes, don't say bacteria. If you find mitochondria, don't say bacteria. That's a no-no. Nucleus, there you go. A cell wall, well, once you see the cell wall, it's not an animal cell. And then, so I'm thinking that what you got to say there, the only one left, golly, it ain't a virus, not a bacteria, it's not an animal. So the only one left is a plant cell. I'd almost want to say fungus, but okay. Little process of elimination. Now I got to move into, like, let's count chromosomes. Woo! Two types of cells in your body as far as chromosomes go. You start thinking about chromosomes or genetic material. We're talking eukaryotic cells. The one type of cell that you got to be concerned about is going to be what is known as the body cell. Okay, body cell. And the other type of cell that you need to be concerned about with the chromosomes is the sex cell. Okay. Let's count up chromosomes in the body cell. Count up chromosomes in the sex cell. Count. Ooh, check it out. The sex cell has 1/2 the number of chromosomes that the body cell does. Now that's strange. Oh, because you get two sex cells together to make the beginning of a new organism, and that new organism has a bunch of body cells, so a half and a half make a whole. So sex cells have 1/2 the chromosomes of the body cells. Keep that in mind. And yes, come on in. Kid just stopped by and visited. I hadn't seen him since last year. We just visited a little bit. I can't remember his name, but nice kid. We got through the conversation okay. Listen, the next thing, we're going to just jump right into it. Did you know that there is a way to mess around with your genes in your cells, especially if they're faulty? And when you start pulling genes out and replacing genes, you're going to start playing the game called recombinant DNA. That's what I'd call it. It may be gene therapy. I think it's recombinant DNA. You better look this one up. Look up the difference between gene therapy and recombinant DNA so that you get that question right. It's the technology which involves replacing an effective gene with a normal gene. That's probably gene therapy. Recombinant DNA, I've got a feeling, is more of a noun, and this question is asking it more about a process. So I'm going to leave that one up to you. Recombinant DNA or gene therapy, find out which one is the process. Okay? Oh my gosh, now we're going into something called transgenic crops. Have you ever heard about that? What they basically do is they take desirable genes and they insert them into agricultural plants. And this may help with the need for water, fertilizer, or may help with reduced need for pesticides. You can like program the plant to produce pesticides of all things. So you get these biotechnologies And I would generally call them genetic engineering when I play around that way. You're like a little genetic engineer. You get to design. Just think, one day you may be designing your own pet. Wouldn't that be cool? Well, I want a dog. I want him 45 pounds. I want the stripes of a tiger. Yeah, I mean, oh, no problem. We'll just insert this gene into this Doberman, take out its fur arrangement, and so I want one. I don't want to have to cut the tail. I want a stubby tail to begin with. Oh, I want a pigtail on it, so we'll just take the gene out. And this at the beginning of the first cell. Oh, wouldn't that be something? Anyway, listen, this is number 2. I'll have to go to number 3, number 4 for you. But have fun. Thank you.